Good morning, students. Today we will be taking a tour through the animal cell. The animal cell is very different from a plant cell. The plant cell holds chloroplast and has a cell wall, while the animal cell has neither. As we arrive to the cell membrane, notice the structures. See all those proteins? This animal must have strong bones. Anyway, the cell membrane regulates what enters and leaves the cell. It also supports and protects the cell. The cell membrane is made up of a lipid bilayer, which has integral and peripheral proteins. The proteins within the membrane can help large or charged particles through the membrane. The other transport that occurs on the cell membrane would be passive transport. This is when small and uncharged particles pass through the cell membrane on their own. Keep moving along. Now we are in the cell itself. Observe all this cytoplasm cushioning the organelles. Now make sure you're paying attention to the free ribosomes floating around. The free ribosomes make proteins for organelles in the cell. To your left is the mitochondria. It looks like the inside of a nutshell to me, but its function is to use energy from food to make high-energy compounds that the cell can use to power growth, development, and movement. Now moving on to the SER, or smooth endoplasmic reticulum. Notice it doesn't have ribosomes on the outside, which is different from the RER, which has the ribosomes on the outside. The function of SER is it collects enzymes that perform specialized tasks and synthesizes of lipids. The smooth endoplasmic reticulum actually has ribosomes on the inside. The centrioles, which look like straws to me, produce microtubules. This is important because microtubules are the main component of the cytoskeleton. The cytoskeleton provides shape to the cell structure. Up ahead is the Golgi apparatus. The Golgi is made up of proteins produced by the RER, or rough endoplasmic reticulum, stacked on top of each other. The Golgi's enzymes attach carbohydrates and lipids to proteins. Vacuoles are to the right of us. This is similar to a storage room. Vacuoles hold water, salts, proteins, carbs, and other materials to keep them out of the way in the cell. Vesicles are the smaller vacuoles that are involved in transporting substances within the cell. Next to the vacuoles, a little up ahead, are the lysosomes. Lysosomes are small organelles filled with enzymes. They break down lipids, carbohydrates, and proteins into particles that can be used for other parts in the cell. As I mentioned earlier, we are coming across the rough endoplasmic reticulum, or RER. The purpose for this organelle is to assemble the cell membrane and some proteins to modify them. Notice the ribosomes on the outside, different from the smooth endoplasmic reticulum. The ribosomes are newly made and become chemically modified. Now for the big finale, the nucleus. The nucleus is the brain of the cell. It controls and contains hereditary information of DNA. In the cell, chromatin is the visible granule material visible. It is spread throughout the nucleus. The nucleus is surrounded by a double membrane called the nuclear envelope. The nuclear envelope has dotted pores which allow materials to move in and out of the nucleus. The nucleus also has a small, dense region called the nucleolus. This is where ribosomes are assembled first. The nucleolus also means little nucleus. Now that we are at the end of this whole animal cell tour, I would like to give an explanation on cell theory. Cell theory is that all living things are composed of cells. They can be unicellular or multicellular. Well, the cell is the basic unit of life, and cells arise from pre-existing cells. I hope you enjoyed my version of the tour of the animal cell.